Hello, everybody, and welcome to another podcast for the U of Care Center of Addiction Research and Education. My name is Oliver Grundmann, and it is my great pleasure to welcome Dr. Robinson from the University of North Carolina. Dr. Robinson, welcome, and do you mind introducing yourself a little bit to our audience?
Sure. Thank you for having me. I'm Donita Robinson. I'm a professor of psychiatry at University of North Carolina in the School of Medicine. And I'm a basic scientist that studies the effect of alcohol on motivation, voluntary behaviors, and kind of motivated behaviors. I'm also associate dean for graduate education here in the School of Medicine. So I'm really involved in biomedical training at all levels.
I'm sure that keeps you quite busy, and it is my understanding from our previous conversation that you just submitted another grant, which is kind of the life cycle of a scientist, right? So
That's right.
congratulations and best of luck and success in getting that funded, hopefully.
Thank you.
Given the your current research, but also being involved across the board with the research that is going on at your College of Medicine, what actually got you initially into addiction science, which might help some of our audience members? We also have graduate students and postdoctoral
Mhm.
fellows seeking kind of where they stand and where they can go in the future. So, do you
Mhm.
mind sharing a little bit of your evolution, so to say, in the field of addiction science?
Yeah, I'm happy to. I kind of call it my scientific story. So I started out in as a psychology major and was just fascinated by the brain and how the brain could impact behavior. And so, in fact, throughout my career, I take that lens of behavior is the ultimate endpoint for all of neuroscience. So, um, but why addiction? Well, I studied motivated behaviors as a undergraduate and then a graduate student. And sometimes I studied, how addiction might and addictive drugs might impact these brain circuits and behaviors. And sometimes I studied them in other contexts, like sexual behavior or maternal behavior. And I was just equally interested in it all. What I'd love to do is put a sensor in the brain and measure what's going on, whether it's neurotransmitters or neural firing patterns, and see what's going on during behavior. And it didn't really matter to me what the context was until I had to write grants. And then when I'm writing grants, I would write grants on different topics, and you kind of see what gets funded. Certainly that kind of propelled me into alcohol research. The alcohol grants got funded. But it's not that simple. It's not just luck, that got me into that. I realized that as I was doing alcohol and other substance use basic science, I realized that so many people, my family, my friends, students, people that I would sit next to on the airplane, their lives had been impacted by addiction and drug use. In lots of different ways. And this is a huge public health issue and a huge societal issue. And I just wanted to be a part of learning about it with the ultimate goal of being able to treat it and help people control their alcohol and drug intake.
That is I think a fascinating story in regards to how behavior is what you said earlier, how behavior is kind of the endpoint of all of these neurochemical and neurophysiological changes occurring, right? And it seems
Mhm.
to be still kind of the holy grail of connecting neurotransmitter release and signaling and what kind of circuits are involved to actually behavior because of the complexity that is involved in it. Now your focus on your current focus, particularly on alcohol addiction or alcohol use disorder, you mentioned funding was one reason why you got into this particular area. But as you kind of are now established very well, in particular the alcohol use disorders,
Mhm.
what do you see in regards to your research and its impact on how we view alcohol use disorder, how we
Mhm.
view behaviors tied to alcohol use disorder, and how this impacts clinical practice?
Right. Well, sometimes I think my basic science doesn't have a big impact on clinical practice. I've got to be honest. And yet that's the driver, that is the driver of why we're doing what we're doing. My current research is very much focused on adolescent alcohol exposure as a model of youth, high school students, maybe even middle school students, but also college students binge drinking, and binge drinking on the weekends, getting drunk, um, as some youth are wont to do, and how that impacts policy. As a college professor, I'm always hearing, often from other professors, that and parents, that maybe the drinking age should be lowered. The drinking age is 21 years old here in the US, but maybe it should be lowered to 18 so that when freshmen and sophomores come to college, they don't drink in their dorm room or in isolation, but rather they would have places where they could drink alcohol in a more supervised setting, and perhaps that would help them to drink more responsibly. Um, that's up for debate. There are data from other countries that might support that don't support that. And so I'm a little agnostic on that end, except if there are long-lasting effects on the brain and on that development that's still going on during adolescence, and you're consuming these high levels of alcohol during that time, I want to know it. And I think that informs our decision on whether we would allow or encourage younger adults to be able to drink when their brain is still developing. And so my lab, along with many other labs, have been focused on this adolescent exposure as a developmental insult. And what we find is that, perhaps not surprisingly, alcohol can act as a teratogen, or a development disruptor in adolescence, just like it can do at other stages of development, like prenatal development. And specifically, what it seems to impact are those prefrontal decision-making parts of the brain and how they interact to control behavior with other parts of the brain. We see that, and these aren't profound effects. Like the animals and people can learn, but there are effects like, when you need to change your behavior for a changing circumstance, how quickly do you do that? How adaptable are you? That ability seems a little blunted. And so I've been looking at animal models of adaptability, of flexibility, and we see that sometimes adolescent alcohol can impact that flexibility long after the last alcohol exposure to the animal. And so I think that has a lot of public health impact. Does it have clinical impact? Well, in our society, binge drinking is not a disease. It's actually, it's like recreation. And it's something that many people do socially. Many people who engage in binge drinking during high school and college do not go on to develop an alcohol use disorder, although it definitely increases the risk of that. And it also increases the risk of maybe trying and being exposed to and having issues with other addictive drugs. So there's definitely a risk there, but in and of itself, it's not addiction. So the clinical impact is a little less clear. Although I do have some happy news, not necessarily from my lab, but from other labs where there are things that can kind of reverse some of these effects of adolescent alcohol exposure. so that if you're, if you're someone of my age who's thinking, what did I do when I was a teenager? You can, and you're thinking, well, how can I increase my behavioral flexibility at this point? One of the things you can do is exercise, good sleep, keeping your immune system healthy. All the things that we know are important for good brain health can also counteract some of these effects of adolescent alcohol exposure. So, so I think that where it's not clinical in the way we might usually think about clinical, as in active treatment for an alcohol use disorder, I think it is healthy behaviors and thinking about how our alcohol use, during adolescence for sure, and actually later in life, moderation's better, less is more in this case. And how we can keep our brains healthy. It definitely has an impact on that.
Thank you for sharing that. And I'm glad that there are good news that we can still potentially even reverse some of the impact that binge drinking during adolescence has. It's kind of interesting that you mention other countries, because I grew up in Germany, where you can drink alcohol, beer and wine. So not the liquors, the hard liquors, but you can drink beer and wine when you're 16. And then with 18, you can drink the harder stuff. I do think there is something enticing, obviously, about drinking when you're not yet of legal age. That seems to propel some of the high risk category folks to engage with it. So when you look at this lower intake of alcohol during adolescence, beer and wine, for example, at 16. And then you look at this binge drinking, high amounts of alcohol that maybe average out to about the same amount over a week or something like that. Do you see changes in animals?
Yeah, so there's definitely something special about binge drinking. Achieving these high levels of alcohol, the concentrations that you have to get drunk, to pass out, have a blackout or a brownout, those are high levels of alcohol in the body, and it's those concentrations that appear to be quite harmful. The binge drinking pattern of achieving these high concentrations periodically, that also seems to be in many cases more detrimental to outcomes than if you were to consume the same amount of alcohol, but over as a more steady intake. So yes, binge drinking is a problem. Even small amounts of drinking or less amounts of drinking, so maybe the amount of maybe one or two standard drinks, during the teenage years, can have measurable impacts in our animal models. There's a set of studies by the late Jeremy Clark, who showed that animals that drank alcohol in Jello, so not surprisingly, animals will consume alcohol in these little Jello shots. And so they were getting about as much alcohol as you would get with one or two standard drinks. They had measurable impacts on impulsivity later in adulthood, that the animals were more impulsive than their counterparts who ate the non-alcoholic Jello. So there's a little less known, though, about these lower concentrations and how they might impact brain circuits and behavior long lasting. Most of the research has focused on binge levels, because we know that that's what's harmful. And if you think about it, that's also, of course, where you have the most risk in the moment, risk for driving accidents, other kinds of accidents, fights and interpersonal issues, and also the consequences around your job and your school and things like that. So there's a lot of immediate impact, and there seems to be a much higher probability of long-term impact with binge drinking.
Now you mentioned earlier that you kind of connecting the dots from an animal model to actual humans, and that you're
Mhm.
working with collaborators together on kind of making these dots a straight line or a filled line. Um, and you're using various different techniques in order to extrapolate to some degree how changes
Mhm.
in animals might also translate to changes in humans, both acutely and ongoing in the long run. Can you talk
Mhm.
a little bit about that and where you at this point?
Yeah, thanks for bringing this up because I think this is a really important part of science, basic science, and also my research in particular. You know, we study basic science many times because we want to improve human health. You know, but we're using these animal models, and sometimes it's hard to understand how it can, you know, how we can relate these things. They're very different, and even the brain of a rat or a mouse is, you know, very different from the brain of a human. Um, but and yet all the parts are there, and so we use these animal models to try to understand what might be happening in people. And when you're studying a complex disease, such as an alcohol use disorder or a substance use disorder, I mean, there's so many things that make it so hard to study in people. um, you, you don't know. So some people may have predispositions to using drugs and alcohol, and those might be personality, they might be genetic, they might be epigenetic, potentially because their parents used alcohol during a critical stage, you know, stage of and they were affected as kids. It might be opportunity because you know, if the alcohol or drugs are available either because they're in the home or they're in the neighborhood, you know, then those kids are more likely to consume than kids where it's not available. So there are many factors that can lead into drug and alcohol use for people. And then once people initiate use, there are the consequences of that use, you know, and so it's hard to disentangle how much of the consequences are actually from the drug versus from these predisposing factors that led them to use the drug in the first place, especially when you're looking at these behavioral endpoints like impulsivity or behavioral flexibility. So we use our animal models strategically to try to answer some of these questions. For example, when I do an animal study where I give the binge levels of alcohol during the adolescent period and only the adolescent period, I'm not necessarily modeling what people do. People don't often binge drink during high school and then stop completely, although a few people do, are you know, do that, but not many. Most people continue to drink, but at different levels later on. You know, but so by using this animal model, what I am saying is asking is whether that exposure, that alcohol exposure, is sufficient to cause some of the behavioral and neural effects that we see in animals and that are observed in people. Now, in order to do this well, I need to collaborate with scientists who study people. And so I'm fortunate to have a longtime collaborator, Charlotte Bodiger, who's here at University of North Carolina. She's in the Department of Psychology and Neuroscience. And she's interested in the same things I am, in behavioral flexibility and adaptability, as well as in those brain circuits that direct our choices and our learning. And she studies this in people. So we figured out about 15 years ago that we were interested in the same things, and we started collaborating. And so we've been fortunate enough to have an NIH-funded grant for the last several years that we work on together. And what that looks like is, you know, she might see something in people who drink. She recruits people who drink a variety of patterns and amounts. And then she can see, oh, when people are binge drinking, they might show this particular phenotype. You know, they might show perseveration on a learned task, even though the rules are changing. And then I can say, okay, how can we model that in our animals and so that we can have a good animal model. So the first step is to have kind of parallel findings. And then the second step, we can, of course, look at molecular or cellular mechanisms, invasive ways to measure circuit activity. We can look at brain slices. We can look at, put sensors directly into the brain. She doesn't have that opportunity, thank goodness. In people, she's using things like MRI and magnetic resonance spectroscopy to be able to get a hint of what kind of chemicals might be in the brain, but it's not a direct measurement, it's an indirect measurement. She can also use transcranial alternating current, so delivering a small current through EEG electrodes to try to change the synchrony and coordination of brain circuits. We can do those things a little more directly. So it's a combination of trying to have parallel translatable findings and then taking each one, the human studies and the animal studies, in the ways that we can to be able to delve deeper. But I do feel like the whole of our research is much more informative than either one would have been on its own, which is awesome, and is why scientists get excited.
I think you make a very interesting, your approach is very interesting, that you first go to humans to observe what is going on, and then you try to match it with an animal model that adequately is able to detect these behavioral changes. And then you digging into the animal model, so to say, and looking at what is going on the neurochemical level, on the cellular or molecular level. So I think that's an interesting approach, because sometimes we try to make an animal model that then kind of mimics what is going on in the human condition, and we don't necessarily know how well that correlates. And here you kind of go back, which I think is really interesting. But it also sounds to me like a lot of these behavioral changes, we know a lot about basic brain circuitry, or that's where we have a good enough understanding. Not a lot is probably not right, because we know at least something about the basic brain circuitry in like the midbrain and in certain striatal regions. But then the prefrontal cortex is still kind of this where the magic happens, so to say, right? And it sounds to me like obviously a lot of behavior is generated or is mediated through prefrontal circuits. So how far do you think you are on that path of linking these pathways to behavior then?
Yeah, and there's, you didn't say it, but there is this big difference between the prefrontal cortex of a human and the prefrontal cortex of a rodent. That's why I say, well, all the parts are there, but they're not as elaborated in the rodent. So certainly there's some limitations to being able to do these trans-species, comparisons. And yet, I think that there's still similarities in that adolescence is when the prefrontal cortex really strengthens its what we call top-down control over subcortical brain circuits. And that happens in mammals. It's not just in humans. We see during adolescence the rise of behaviors that we think of as being human adolescent behaviors, things like risk-taking behavior, sensation-seeking, preferring peer interactions over parental interactions. All of those things actually happen in rats and mice and puppies, so this is a mammalian thing and not in a biological thing. That's actually quite adaptive. So I think it is a good model to be able to figure out, back and forth. But, prefrontal cortex is it is more important in humans than it is in rodents if you think about real estate of the brain. And so there might be some limitations that we have, but it's still worth a try. The prefrontal cortex is relatively new for my career. As a true basic scientist, I started, in a different brain region, and for me, it was the nucleus accumbens, and I ignored the rest of the brain, for many years. And then only gradually have I learned to kind of expand the circuit as I have become more and more interested in particular questions and in my case, in behavioral flexibility.
I'm sure that many parents appreciate you saying that it's not specific to humans, that peer interactions are sought over parental interactions in adolescence, because that is when tensions are rising, let's say it that way, during adolescence. I'm glad that also happens in other species.
Oh,
So
yeah.
glad to hear that. So in regards to where you see your research going in the next years, what do you think are you kind of addressed some of that already, the translational aspect. Where do you think are opportunities for you in the near future, in the short term, in the long term?
For in the relative short term, my lab is going to be looking at some of these, some neurochemical and kind of structural changes in the brain that might be affected by adolescent alcohol exposure, and might explain some of or at least contribute to the behavioral effects that we're seeing, and some of the brain connectivity and brain network effects that we're seeing. And specifically, these are we're interested in a particular neuron in the prefrontal cortex. It's an interneuron. It's GABAergic and fast-firing interneuron that expresses parvalbumin. And these parvalbumin positive neurons are known for kind of coordinating network activity. They're important in synchronizing activity within a brain region and across regions. And so we think they're a good target for looking at why connectivity might be disrupted when we use something like resting state functional connectivity, MRI, to look at the effects of adolescent alcohol exposure. So we're targeting those neurons. Those neurons are often surrounded in the tissue by these extracellular matrix structures called perineuronal nets. And the perineuronal nets are interesting because when rats or individuals are really young, there aren't very many perineuronal nets, if any, and they start to grow during development, and especially in the adolescent period. And so then as they reach adult levels, they are thought to, maybe close a window of plasticity. And so if alcohol is impacting the deposition of these extracellular matrix structures around these important neurons, it might be prematurely capturing these neurons at an adolescent state, which could explain, now I'm speculating here, just to be clear, but it could explain some of the adolescent phenotypes that we see persisting into adulthood after alcohol exposure, like higher impulsivity, distractibility, and things like that. So that's one of the kind of, in the short term, that's where our lab is going. In the long term, I just see myself continuing to be interested in translation, in making sure that our animal models are modeling things that are happening in people. That just requires constant vigilance and learning, learning about what's going on in the clinic, what's happening, in my collaborators' labs, but in the broader field. There are a lot of rich data sets that are being built now, especially developmental data sets. There's a large study, the ABCD study, that's funded by the NIH to study adolescent development in several thousand individuals. And they'll be tracking a lot of things, including drug and alcohol use and things like that. But before that study started, that study's pretty well known. There was a study funded by the National Institute of Alcoholism and Alcohol Use in IAAA that did a very similar study, but really focused on alcohol use, and that consortium's called INCANDA, and they've been funded for over 10 years, I think more like 15 years. And so they're really getting data now on both the antecedents of binge drinking in adolescents, but also the consequences. And so, kind of looking at those longitudinal human studies that are so difficult to do, but so important, and then making sure, using our animal models strategically to be able to say, okay, we see this happening in people, we can replicate it in our animals. What's really going on at a cellular circuit level that we can maybe, target to improve health in people. And so going back and forth between the human and animal studies, I don't think I'll ever get tired of that.
Well, considering the complexity of the human brain and how little we understand, I think there's plenty of still research that can elucidate how behavior is based on potentially this initial formation of these extra neuronal networks, or that kind of form personality traits, or certain
maybe.
behavioral patterns, and if that's disrupted, then you get impulsivity. That, I mean, it makes sense. You said you're speculating, but I think there are, on a larger scale, it does make sense. As we go through adolescence, certain behavioral patterns kind of establish themselves and really settle in, and then maybe that's disrupted. So that's a great approach. It does make sense. So what do you think are, to conclude kind of this podcast, what do you think are the major obstacles you and other researchers when it comes to alcohol use disorder, but addiction science in general? What do you face? What do you see as major obstacles that are still potentially keeping you from really achieving what you want to achieve, or the next step in, or maybe on a societal basis as well? What are some of the major obstacles?
Well, let me just pick a few. So with research on alcohol use, substance use, and a variety of use disorders, as well as non-drug disorders, disorders of behavior, and addiction is kind of, that whole word is kind of going out of favor for trying to be more explicit and specific, right? But there's still so much stigma. There's a lot of stigma around it that impacts how we as a society view the importance of this work, and impacts the amount of compassion that we have when we view patients and people who are dealing with these disorders. So that's a barrier. I think there's even a more general barrier of funding in science, that our society values science. We love being at the top, internationally, in scientific discoveries, but we're not putting the resources behind it that would keep us there. And you see that, and there's also, I think, some challenges on the biochemical workforce, and getting. I don't think we have a problem getting people interested in doing research, but we have a problem retaining the best and brightest minds, because it's not necessarily a lucrative career. There are other career paths that might, be easier, more pleasant, and pay better. So I think, I'm very interested in making academics and research more broadly an attractive career option, improving the quality of our workplaces, And I firmly believe that those efforts will also increase the quality of the science that we produce and the discoveries that we make.
In regards to the stigma surrounding substance use disorders or use disorders in general, I think every single podcast interview that I conducted with other folks working in this field, they have pointed this out. And it speaks to us as a society that this dirty word addiction has so many negative connotations associated with it that we can't get rid of, it looks like. And so shifting to defining it as a disease, as a disorder, is hopefully settling in with future generations to really look at this from a very different perspective. So I commend you for definitely working to destigmatize some of the issues that are specifically associated with alcohol, but substance use disorder in general. In regards to the academic work environment and compensation and all of that, I think we are luckily moving in the direction where we find that diversity and equality within that space matter very much, potentially even more so than just reaching an industry level pay grade. I think it's primarily passion driven. Like it comes through in everything you say. You're passionate about this, but I think it's also, if we create a work environment where folks feel welcome as they are, I think that already makes a huge difference in regards to welcoming women, minority folks of all walks of life that bring their own experience, their own lived experiences to it is definitely quite impactful.
Absolutely. Yeah. Absolutely. And
Well,
there's a lot of research to back it up that,
yeah.
People who diverse teams are more productive. They're more creative. They're more productive. And they just do better. And so we would, our networks are often filled with people who are like us. But if we can reach beyond that, we'll be better scientists for sure. And part of that is exactly what you said. Everybody needs to feel safe, supported, and excited to be in their lab and in their workplace. And that's my goal.
And this is a wonderful way to end this podcast. I really appreciate you, Dr. Robinson, for joining us today. And I cannot wait for future research to come out of your lab and out of your collaborations that really will inform us in regards to behavior resulting from substance use disorder, and in particular in the alcohol field. Thank you so much for spending time with us today.
Thank you. Thank you for having me. It was a pleasure.
Thank you to everybody listening to this podcast. Please feel free to share this podcast with your network and also outside of your network. Post it to social media so that we can spread the word and can inform the general public as well as the scientific community. See you at the next podcast.